this catheter here. It's a special type of blood pressure catheter called a solid state catheter, and what that means is this will sit inside an artery. So in our animal model, we implant this into the abdominal aorta. So we just make a little hole, slip this in, and kind of glue it in place, and that can sit in there for months and record pressure. The solid state aspect means that the pressure signal is transduced at the very tip of this. So a traditional way to measure blood pressure is to use a fluid-filled catheter, where the pressure signal would have to be carried all the way along on the catheter to the body of the telemeter, or outside the animal if it was a catheter running outside the animal. But that creates an opportunity for noise or movement artifact to get into that signal. So by picking up the signal and transducing it here, that means we're recording exactly what's happening. So it's the most high resolution signal that you can currently get. If you've been used to working with the fluid-filled catheters and you switch to the solid state, you'll notice what you're seeing on the screen looks a little bit different. That's actually real. So what you're recording with these solid state catheters is actually a better reflection of what's happening sort of inside the animal. You see usually a higher systolic, kind of a pointier angle, and you see a bit more dynamic change throughout the pulse cycle. So you'll see a little bit more of a bump depending on where you're recording after the systolic peak. If you're just looking at mean, it's not critical because, over longer periods of time, the mean level of pressure will be similar. But if you're interested in looking at sort of diastolic and systolic, the systolic in particular tends to be underestimated by the fluid-filled catheters because they tend to damp that signal down. The other problem can be if you're looking in other beds. So as part of our stroke studies, we my PhD student did some fairly heroic studies where he recorded blood pressure, intracranial pressure, and brain tissue oxygen. So simultaneously in rats, instrumented them, recovered them, recorded a baseline, induced a stroke, then recorded afterwards. Intracranial pressure is an interesting one because the pressure in the brain is very low. So in the order of usually between 4 and 8 millimeters of mercury, so a lot less than arterial pressure. And because that's quite low, it's actually, the pressure will change with animal posture. So if the animal rears up, there's a true change in ICP. If you've got a fluid-filled catheter, because the signals are transduced at the body, you tend to underestimate some of those changes because if the animal's upright, the fluid-filled catheter is upright relative to the body, and you don't kind of see that response. When you've got the transduction happening at the tip, all of those positional changes, which is actually what the brain is experiencing at those times, will be reflected in the pressure that you're recording. So it's completely possible for intracranial pressure to go negative. And in fact, it probably happens quite a lot because you think about when you've been lying down, your body gets into equilibrium and you suddenly spring up, particularly in larger animals like humans where the size of that gap is bigger. Your intracranial pressure can go negative and then it'll kind of adjust and come back up. There's various reasons why these solid state catheters really, they're kind of worth their weight in gold.